Hey everyone, we are back with cell structure. A lot of you guys indicated last year this is your least favorite topic, but we still have to learn it anyway. So chapter 6 is cell structure. We have pictures of cells here. Alright, so cells all basically have the same characteristics. They all have genetic material. They could be single circular molecule located near the center of this, that says call, but it should be cell in prokaryotes, I apologize. So in the center of the cell in prokaryotes, hopefully you have the volume on. There's a double helix located in the nucleus of eukaryotes. So we have two different types of cells, prokaryotes, eukaryotes. We're going to be talking about both types today. All cells have cytoplasm that fills the cell interior. And all cells have a plasma membrane that encloses the cell and separates the cell from its surroundings and basically allows things in and out. Alright, so this is a generalized eukaryotic cell. We are going to learn about prokaryotic as well, but we take a look and we see that we have our plasma membrane, the cytoplasm, organelles. All eukaryotic cells have organelles, however prokaryotic do not have membrane-bound organelles. A nuclear envelope and a nucleus. Again, prokaryotes do not have that, but we'll cover that more later. We need to first talk about the cell theory. The first part of the cell theory is that all organisms are composed of one or more cells, and the life processes of metabolism and heredity occur within these cells. Metabolism, we're going to be talking a lot about this year, but all metabolism really means for our purposes today is all the reactions that go on. They occur inside of cells. And heredity, obviously, is dealing with our DNA. 2. Cells are the smallest living units of all living organisms. Now, we're not saying that cells are smaller than atoms. We're saying that they are the smallest living unit of all living organisms. They're made up of organelles, which are in turn made up of molecules, which are in turn made up of atoms. But these are the first. Cells arise only by the division of a previously existing cell, which means cells just can't pop out of thin air. They have to come from another cell. Very good. Moving on. Cell size. Monday we're going to be doing a lab to explore this concept a little bit more. But small cells have a high surface area to volume ratio. An increase in surface area in small cells is an advantage for the cell. Why might that be? All substances enter and exit through the plasma membrane. It provides the cell with more control of what exits and enters. This is just a little graphic to show you total surface area of these boxes, I guess. So if we take a look, I have a cursor on this, I forgot. This has the advantage, okay? The surface to volume ratio is our surface area divided by the volume. We're going to be working on this together. So which one has the highest? These guys do. They are at more of an advantage. All right, so visualizing cells, we do need to talk about how we know cells are, how do we know that we have these cells? And of course, like all science, it starts off with a really bored man. And there was a scientist who decided to slice a piece of cork and look at it under the first rudimentary light microscope, and he saw that it looked like little compartments. He called them cells right next to each other. So there we had it. We use microscopes to observe cells. Now we use a light microscope, or we actually use a light compound microscope. We look at different lenses. There's a transmission electron microscope. Electrons are transmitted through the microscope, which means, because you all remember that atoms are the smallest component to matter, electrons are even smaller, so an electron microscope allows us to see things that are very, very tiny, all the way down to the smallest virus, which are fairly small. A scanning electron microscope, same thing. We can see small things. We'll be using light microscopes in our lab to observe cells. However, we're not going to be able to see all the different organelles of the cells like we would be able to if we were looking at an electron microscope. So we take a look at the things that we can see. The human eye can see a paramecium, human egg, adult human, okay. These are the things that we can see. Let me take a look at the light microscope, which are things that we can look at. So we can look at a paramecium under the microscope, a human egg, a blood cell, a bacteria, several other different cells. But then we go over to this electron microscope and we see they can see much smaller things. So we're looking at a virus, we're looking at a ribosome, we're looking at an actual protein, even further down an amino acid, and even further still an hydrogen atom, which is pretty impressive. It would be cool to have an electron microscope, but I don't have like 2 mil hanging around to buy one. All right, prokaryotic cells. So there are two types of cells. We have eukaryotic and prokaryotic. We'll talk about prokaryotic first. They're the simplest organisms, and they are unicellular, one cell. The most common unicellular organism is a bacteria. However, they are not the only unicellular organism. We also have yeast, which is a unicellular fungus. Okay, it's very important to the economy of living organisms. Prokaryotic cells have a cytoplasm surrounded by a plasma membrane and encased in a rigid cell wall composed of peptidoglycan. The cell wall of prokaryotes is very important for a few reasons. They have no distinct interior compartments and can have two different types of cell walls. The first is called gram-positive, a thick, single-layered cell wall that retains a violet dye. There's a test that we do called the gram staining test that determines what type of cell wall we have. This comes in handy, and I'll explain it in just a minute. Gram-negative is a multi-layered cell wall that does not retain the purple dye but turns red instead. So scientists will do this test. They'll see that it turns purple or red, and we'll know we have a gram-positive or a gram-negative bacteria. This is really important for us because we need to know what type of antibiotics to use to kill a bacterial infection. So when you go on antibiotics, the reason why the bacterial cell is killed is because the cell wall is split open and everything comes gushing out. So we need to know how to attack that cell wall. If it's a gram-positive, it doesn't need to be a super strong antibiotic, but then we have gram-negative, we know that we have a thicker multi-layered wall that we need to get through. Yeah, there we go. All right. So here's a picture of a generalized prokaryotic cell. Some of them use what's called a flagellum for locomotion. It's a thread-like structure that protrudes from the cell and it helps it move. So we take a look here. I have another picture after this. Here's our there's a little motor here that moves this flagella, and this is the flagella. And these are little pili. Ribosomes. There are ribosomes in prokaryotic cells, and think about why. If we have DNA, we have to have ribosomes because of the product of what DNA is coding for. Please also note, and a lot of people get really confused with this, there is a cell membrane along with a cell wall. And this pink area out here is what we call a capsule. Here's another picture. It's showing you our bacterial chromosome. We call it a nucleoid. It's just a generalized region in the middle of the cell. This one happens to have 3 flagellum. Cell wall, cell membrane. Okay. And it's a typical rod-shaped bacteria. Last year I taught you guys about the different shapes. Bacteria can be circular, rod-shaped, or spiral. All right, here we are to eukaryotic cells, and we're going to spend the majority of our time here. They're characterized by compartmentalization from an endomembrane system. Oof, that was a mouthful. And the presence of membrane-bound organelles. The plasma membrane, we'll start off talking about that. It's going to be a brief discussion because we get into the plasma membrane. It's its own topic. It's a selective barrier that allows sufficient passage of oxygen, nutrients, and waste to service the volume of every cell. The general structure of a biological membrane is a double layer of phospholipids. You should know what that is. We see lipid in there, we know fat, we know doesn't like water. But then you see the phosphate group on there, and we know that phosphates do like water. So you have a phospholipid. Let's take a look. We have our phospholipid. This is our phosphate head. Here's our lipid fatty acid tails. I don't know if you guys remember that. I'll stop saying I don't know if you remember. I'm going to assume that we don't remember. So this is our fatty acid tails. We have the fats in the middle and the hydrophilic heads facing the water. That's how they arrange themselves. Proteins injected into the cell membrane, some carbohydrate side chains, all as well. The nucleus. The nucleus is the location of our genetic material. Inside of the nucleus is a nucleolus. It's a region of intensive ribosomal RNA synthesis. Essentially, this is where our ribosomes are put together. The surface of the nucleus is bound by two phospholipid bilayer membranes. We call it the nuclear membrane or the nuclear envelope. So here's a picture. We see our animal cell and we take the nucleus out. The nucleus also has nuclear pores. Why might that be? What needs to exit and enter the nucleus? Here's our nucleolus and our nuclear envelope or nuclear membrane. The stuff inside of the nucleus, instead of cytoplasm, is called nucleoplasm. Here's our inner membrane and our outer membrane. Just another picture to show you. We have our nucleolus. You see a lot of chromatin in here. Nuclear envelope, inner membrane, outer membrane. We have pores for molecules like RNA to come in and leave. Why does RNA need to come in and leave? Great question, right? And here's a close-up of the nuclear envelope, and here's our chromatin, and we're going to talk about that in just a minute. So chromosomes is the DNA of eukaryotes is divided into linear chromosomes. They exist as strands of chromatin, except during cell division. When the cell is functioning normally, DNA is loosely wrapped around histones, which are proteins. Packaging proteins, thank you. Nucleosomes are clusters of histones. I'm going to show you a picture so you understand. Here's our chromosome. We all know what a chromosome looks like. If we take a closer look, we see that these pink balls are the histones, these proteins that allow for the chromatin to wrap, the DNA to wrap around these little packaging balls to give it more room. So the blue is the DNA, the pink is our histones, we have spacer histones, the whole thing is called a nucleosome. Alright, we're going to talk about our endomembrane system now. I'm struggling today to talk, I'm sorry. Compartmentalizes the cell, channeling passage of molecules through the cell's interior. Provides surfaces for the synthesis of lipids and some proteins. We have the endoplasmic reticulum. The rough ER is studded with ribosomes. It manufactures proteins for export. Devoted to protein synthesis. Proteins are manufactured on the surface of the rough ER and enter through the membrane of the ER into the cisternal space. The smooth ER has very few ribosomes, if any. Membranes contain many embedded enzymes. Enzymes catalyze the synthesis of carbohydrates and lipids. So let's take a look here. We have our smooth ER, we have our rough ER. The cisternal space, I know I said that word earlier and you probably are like, what does that word mean? We're talking about the space inside of the membranes, the cisternal space. So you have ribosomes on the rough ER and embedded here, and then in our smooth ER there's a lot of enzymes embedded into the membrane, and it's just what it looks like under a microscope. Alright, the Golgi apparatus. Believe it or not, the Golgi apparatus is very complex, so we're going to spend a couple minutes talking about this. It's a collection of Golgi bodies. They collect, package, and distribute molecules synthesized at one location in the cell and used at another location. So materials made all over the cell go to the Golgi apparatus to be modified and then get shipped out to be used somewhere else. It has a front and a back with a different membrane composition, and this is really important. The front we call the cis face. It's located near the ER, endoplasmic reticulum. Materials move towards the cis face in transport vesicles that bud off of the ER. The vesicles fuse with the cis face, emptying their contents into the interior of the Golgi apparatus. We call it the lumen. The back of the Golgi apparatus we call the trans face. After the material has passed through the Golgi apparatus, it is discharged into secretory vessels through the trans face. So here's a picture of our Golgi apparatus. Cis face is the front, trans face is the back. And these are transport vesicles that just come together and fuse with the cis face and then are released from the back in the trans face. I'll have better pictures in just a minute. Bear with me. All right, proteins and lipids are manufactured in the ER, the rough and the smooth, the endoplasmic reticulum. They're transported to the Golgi apparatus. The Golgi apparatus modifies them as they pass through. The most common modification is adding a short sugar chain, carbohydrate. The modified substance travels to the cisternae, which is the ends of the Golgi apparatus, and then they're pushed out in secretory vesicles. Let's take a look. Yes, here we go. One of my favorite pictures. You will see this picture on a test. I promise, promise, promise. So let's go ahead and take a look at what we have going on here. Here we have the rough ER. The rough ER is where proteins primarily are made. So let's call these little purple things proteins. So they get to the end of the rough ER, and what happens is they form a little bulge around them and pop the proteins right out. So we have a transport vesicle surrounding them so the lysosomes don't come and digest them. So this transport vesicle then fuses with the cis face of the Golgi apparatus. The Golgi apparatus modifies the proteins, adds carbohydrates, does whatever it needs to do to it. It travels through the cisternae of the Golgi apparatus until it is then pushed out the back. It's modified now in these little secretory vesicles. Again, these protect them from being killed by the lysosomes or digested. And then this vesicle just fuses with the cell membrane and releases its contents into the extracellular fluid. And then this macromolecule goes where it needs to go. Pretty cool, isn't it? Again, this is just a better picture. We have proteins, we have transport, cis, okay, great, lovely. Migrating transport vesicle. All right, we're still on the endomembrane system. Vesicles. Lysosomes are the ones that we know the best. Remember, because I made you play that game and you guys hated it, but I loved it, and the lysosomes would come eat you guys. The membrane-bound digestive vesicles. They contain many enzymes that go around digesting various molecules. They arise from the Golgi apparatus. Okay? They contain degrading enzymes or digestive enzymes that catalyze the breakdown of proteins, nucleic acids, lipids, and carbohydrates. We also have something called microbodies, and these are enzyme-bearing membrane-enclosed vesicles. Peroxisomes are the ones that we see the most. They contain enzymes that catalyze the removal of electrons and associated hydrogen ions. That will make more sense once we get into cellular respiration and you understand why we have extra electrons and hydrogen atoms hanging around. This is showing us a picture of, okay, let's take a look here. So here we have this phagocytosis is cell eating, essentially. We have a food vesicle, nutrients. You see a lysosome budding off of the Golgi apparatus. It comes and fuses and digests all the food particles for the cell. We call it food particles, but really it is some form of nutrition for the cell. Cells need to eat too. Then we take a look. It also works on old or damaged organelles. So the lysosomes bud off the Golgi apparatus. You see them come engulf this old yucky mitochondria and it breaks it down. Ribosomes, one of my favorite organelles. They're so cool looking. So when you learn them before, they look like these little dots. But really, they have two subunits, a large subunit and a small subunit. Ribosomes are RNA protein complexes. They're composed of two subunits that join and attach to messenger RNA, and they're the site of protein synthesis. So if you remember, it attaches to the mRNA, which is only one line, and it reads the code by moving down the line and attaching amino acids together through dehydration synthesis to make a polypeptide. Oof. These ribosomes are made in the nucleolus. So organelles with DNA. The mitochondria, let's talk about that one first. It's bounded by an exterior and interior membrane. The interior is partitioned off by what we call cristae. It's where cellular respiration occurs, and what we should know basically right now is that cellular respiration results in energy, ATP, by using oxygen and sugar. It acts as a cell within a cell. They divide themselves inside of the cell, and they have their own DNA, which is really interesting, and we're going to talk about theories behind that. The chloroplasts, which are not in animal cells, but they are in plant cells, we're going to talk about them here. They have an enclosed internal compartments of stacked grana that we call thylakoids. They're found in photosynthetic organisms only and can manufacture their own food. So we're taking a look at a picture of the mitochondria. Here is our chloroplast. We have our grana, our thylakoids. This is the site of photosynthesis. So there's an evolutionary theory about endosymbiosis. The endosymbiotic theory suggests that engulfed prokaryotes provided hosts with an advantage. So let's just call this vague picture, this is a prokaryote, this is our chloroplast. And what was thought is that it was an evolutionary advantage for these prokaryotes to engulf the chloroplast, same with the mitochondria, so that one, they could make their own food, and two, they could have energy. The cytoskeleton, okay, it's a network of protein fibers supporting the cell shape and anchoring organelles. We have actin filaments, okay, which have to do with cell movement. Actin is a protein. We have microtubules, which come in the form of centrioles. Centrioles will play a very important role in cellular division. And then intermediate filaments. Oh, guess there's nothing there. Let's take a look at the intermediate filaments, the microtubules, and the actin. So when we learn about muscles, you'll learn a lot more about actin and myosin. But for now, we need to know that they stabilize the cell and they act as a skeleton. In this table, it's just showing us the structure and function of the cytoskeleton. So microtubules are hollow tubes. They maintain the cell shape. They help cells move. They chromosome movements and cell division as well as organelle movement. Then we go over and look at microfilaments. These are actin. They maintain cell shape. They change cell shape. They have to do muscle contraction and cell movement and cell division. And then we have our intermediate filaments, one of many proteins. They maintain cell shape. They anchor the nucleus and other organelles, and they, that's what's important to us. All right, so really quickly, animal cells, we need to address this. Animal cells lack cell walls, we know this. They form what's called an extracellular matrix. So the word extra means outside of the cell matrix. It provides support, strength, and resilience for the animal cell because they don't have that cell wall. Oh, yes. Let's play this. Oh, please work.
And check it out. We've arrived in the space between two cells. A sticky coat called the extracellular matrix holds the cells together. Each cell is surrounded by a flexible plasma membrane with an incredible number of projections, docking stations, and channels. Let's dive into one of these channels to enter a cell. Whoa, look at this place. These girders and cables make up the cytoskeleton, the structural framework of the cell. They also serve as tracks for transporting cargo from one place to another. All this activity in the cell requires energy in the form of ATP molecules, which are made here in the mitochondrion. Notice the outer membrane and the inner membrane with its numerous infoldings. Many of the molecules involved in making ATP are built into the inner membrane. All those folds increase the inner surface area, enabling more ATP to be made. Moving towards the nucleus, we pass by layers of internal membranes. The nucleus is enclosed by a double membrane called the nuclear envelope. Let's enter the nucleus through a pore. The nucleus houses the genetic material of the cell, DNA, which carries the blueprints for making the cell's proteins. Almost 2 meters of DNA is crammed inside the nucleus. How does it all fit? The DNA is wrapped around proteins like thread wrapped around spools. Look, This section of the DNA has unwound, and a different protein has attached to the DNA. DNA is being used as a template to make mRNA. mRNA molecules travel from the nucleus to the cytoplasm, carrying the instructions for making specific proteins. In the cytoplasm, a ribosome clamps onto a strand of mRNA. The ribosome ratchets along the mRNA, building a new protein. Some proteins stay in the cytoplasm. Others, like this one, are processed in special compartments within the cell. Protein processing and certain other metabolic activities occur in the endomembrane system, the cell's network of internal membranes. The endoplasmic reticulum, or ER, is part of the endomembrane system. There are two types of ER, rough and smooth. Rough ER is covered with ribosomes. Smooth ER lacks ribosomes. Lipids are made in the smooth ER. Let's go inside the rough ER. Note that you can still see the ribosome on the outside surface. The ribosome is manufacturing a new protein, which continues to grow inside the ER. Completed proteins move to the edge of the rough ER and depart in a vesicle that buds off from the ER membrane. Some vesicles fuse with the Golgi apparatus, another component of the endomembrane system. In the Golgi, proteins undergo further processing. Finished proteins are then packaged in vesicles that pinch off from the Golgi and are transported along cytoskeleton tracks. Some vesicles bind with the plasma membrane, secreting their contents outside the cell. Other vesicles, called lysosomes, contain digestive enzymes. Here, a lysosome fuses with a worn-out mitochondrion and breaks it down. Each of the trillions of cells in your body is a dynamo of activity, requiring millions of ATPs every minute. But most people are unaware of all this activity in their cells.
All right, interesting stuff. Hold on, sorry, let me get back to my PowerPoint. Okay. So, yes, tour of an animal cell. Here's a picture of an animal cell. You guys will be required to know and understand where all of these organelles are and what they do. We'll do some things to help you with that. All right, so let's talk about plant cells. Plant cells have all the same organelles as animal cells, except for they have 3 extra. And let's talk about those. We have the central vacuole, which is a large compartment found in the center of a plant. It serves as a storage facility for water and other materials. The central vacuole has a lot to do with the turbidity of plant cells, which we will be exploring later on, and holding all that water and keeping plants with their shape. The cell wall we are going to talk about in detail after this slide. It's a primary wall. Oh, it's got several. I'm going to move on. It's got several components. So let's talk about the cell wall. The cell wall is an extracellular structure that distinguishes plant cells from animal cells. Prokaryotes, fungi, and some protists also have cell walls. The cell wall protects the plant cell, maintains its shape, and prevents excessive uptake of water. Plant cell walls are made of cellulose fibers embedded in other polysaccharides and proteins. So you see the word saccharide, you know sugar, you know carbohydrate, many carbohydrates, and protein. The plant cell walls may have multiple layers. We have the primary cell wall, which is relatively thin and flexible. We have a middle lamella, a thin layer between primary walls of adjacent cells. And you have a secondary cell wall, only in some cells, added between the plasma membrane and the primary cell wall. I'm hoping that tomorrow, since you guys should be watching this Tuesday night, you will have the opportunity to see this. The plasmodesmata are channels between adjacent plant cells. So we're taking a look at the picture here. We have our secondary cell wall, our primary cell wall on the middle lamella. Same picture. I have another awesome show for you to watch. Hold on.
Animals and plants are made up of cells. Their cells have many features in common, but there are a few significant differences. Let's look inside a leaf to take a closer look at a plant cell. First, we encounter a protective cell wall outside the plasma membrane. The cell wall is made from strong cellulose fibrils. Once inside the plant cell, we see the large central vacuole, which regulates the composition of the cytoplasm, creates the internal pressure that is characteristic of plant cells, and stores various compounds produced by the cell. Plants make their own food by photosynthesis in chloroplasts. Light passes through the two membranes of the chloroplast and strikes these green discs, where light energy is converted to chemical energy. The sugar molecules produced by photosynthesis can be made into other molecules or broken down for energy. All plant cells have mitochondria, just like animal cells do. Sugars produced by photosynthesis are broken down and converted to ATP in mitochondria. Most organelles, like mitochondria, are found in both plant cells and animal cells. So the next time you pass by a plant, remember that we have more in common than meets the eye.
So exciting. All right, we're almost finished here. A picture of our animal cell. There's our plant cell.
Both animals and plants are made up of cells.
Hold on.
Their cells have many features in common, but there are a few significant
Nope, that's not what I need.
differences. Let's look inside a leaf
Let me
to
pause
take
this. Okay, I apologize. Here we go. Last thing we need to talk about in this lecture, and I know it's been long, so great job hanging in there. Cell junctions. Okay, neighboring cells and tissues, organs, and organ systems often adhere, interact, and communicate through direct physical contact, and we're going to actually spend a lot of time talking about that. The intercellular junctions facilitate this contact. There are several types of intercellular junctions. We have a plasmodesmata, which we saw earlier, a tight junction, and desmosomes, gap junctions as well. So really quick, these plasmodesmata are channels that perforate plant cell walls. Water and small solutes, sometimes proteins and RNA, can pass from cell to cell. The tight junctions, the desmosomes, and the gap junctions in animal cells. Tight junctions, membranes of neighboring cells are pressed together, preventing the leakage of extracellular fluid. The desmosomes are anchoring junctions. They fasten cells together in strong sheets. And the gap junctions are like our communicators. They provide cytoplasmic channels between adjacent cells. You are going to have to know the difference between all of these. Let's take a look at this picture. We have a tight junction right here, holding the cell together, a desmosome, and then our gap junction actually connecting cytoplasm to cytoplasm. This is just a table showing you the difference in prokaryotic animal and plant cells, the description on the function of all the cell organelles, and that'll do it. This is a nice picture of an onion and mitosis. Looks like metaphase. So that's it.